morning. Another Friday. Cheers to the weekend. There was some random name on there that I'm not familiar with, so I just removed them. Hopefully not a Zoom bomber. You guys doing okay? Good. All right. Well, morning, everybody. Happy Friday. I'm going to share screen so we can kind of get the logistical stuff out of the way. Okay, I wanted to just pull up our Moodle syllabus, make sure you guys saw the daily schedule, the tabular form. And again, reminder, exam 1 is going to start, be open and available on Wednesday, the 24th. So we won't be meeting, so you can take the test during our class time, but I'll leave it open for 24 hours and give you guys that 1 hour time period for whenever you're ready. So in case people are trying to plan it around work or trying to get back from an in-person class to their space, you guys will have that opportunity to do that. And the format will be the same, right? And content will probably cover through plants. All right, any questions? All right. Additionally, just looking ahead, we'll be shifting over into animals after our plant lecture. And then we'll have life history and demography. The demography is going to be easy for you guys now that we've had lab. It usually kind of goes in opposite order in other years. And genetics, and then population growth. So this next unit has, notice in blue here, it does have a homework assignment, a brief homework assignment for each one of those. Mostly it's for reinforcement of math, right? So there'll be math for the demography, math for the genetics, math for the population growth. The life history one is just kind of a fun drawing one, so you can kind of use your creative side for that. But again, try to, when we cover the content in lecture, try and within the next, lecture or two, get to that homework assignment so it serves as reinforcement while it's fresh in your mind, okay? I'll accept those homework assignments throughout, but if you can get to them kind of really, within the next lecture or two, that would be ideal for your learning, okay? So I just want to make sure you're prepared for that. They're already posted, and you'll be able to start them, once we've covered that content, okay? All right, let's begin our lecture. We're on plants today, and I have a lot that I want to cover with you guys. Next week in lab, we're Zooming, okay? So you're going to be remote again, and we'll be basically, looking at the squirrel mapper stuff. So feel free to review that over the weekend so you're comfortable with the material. All right, so we started off with plant adaptations. And we had begun kind of just talking about, the energy the plant usually is using to kind of catalyze their own growth, survival and reproduction, is coming from the sunlight, which is striking the earth or, at either on land or in, an aquatic scenario. And we mentioned there's a lot of impediments that it'll pass as it strikes the earth, refracting back from the clouds and so on. So, think about the breakdown of land. 30% of the globe is land, right? And we've got our plants growing in these terrestrial environments. They're going to be pulling in carbon dioxide from the air and nutrients and water from the soil in order for them to meet their metabolic demands and photosynthesize. So on the Moodle overview page, there's kind of an organization that I've put together. We're talking about plants broadly, and we're talking about their adaptations. So I've kind of broken the lecture it's broken out in sections. So you're going to have, plant adaptations to, physiological adaptations. So how they're metabolizing. So we'll think about, C3, C4, and CAM metabolism in order for them to kind of, convert things to sugar. And then we're going to talk about them morphologically. So the shapes of their leaves, kind of the location of their stomates, how deep their roots are, that sort of thing. We've covered a little bit of that in our earlier classes. So anyways, this is sort of an overview, right? We're going to think about how plants will access nutrients. We'll think about how they will photosynthesize or their metabolism, which will be either C3, C4, or CAM pathways, CAM being the desert plants. We'll talk about how these organisms respire, so how they'll burn that sugar once they've made it in order to kind of grow and do their normal functioning. We'll talk about the key structures in plants, the stomates, which we've talked about before, but these are the gas exchanging pores in most of these plants. So they'll open up to take in carbon dioxide, and they'll at that same time be able to release some of the water. If it's windy or if it's dry out that day, water will get pulled out of those stomates. So these plants really have to kind of balance how they're going to deal with that, right? It's risky if you're in a hot scenario, like you're in the desert and you go to open your stomates to take in carbon dioxide to make sugar because you need to live, right? But at the same time, you're going to lose that precious water. And you've all seen probably plants that wilt, right? If I forget to water my hydroponic basil, it's going to be in trouble in a couple days, but then you can, add water to it and it perks right back up. And so we'll talk about those features. And so, these are the structural adaptations, how the stomates will work in different biomes, right? All that stuff is things we'll cover. So, these plants, they have unique traits, again, whether it's their metabolism or whether it's their shape, and those traits will vary depending on their environment. So I threw this slide in there and talked about it last time that, if a plant is located in a wet area, it's going to have certain adaptations that are not going to be really useful for them, maybe in a dry area. So, think about how habitats can shift dramatically, right? We talked about El Nino. You might have a drought one year, you could have, extreme rains in another year, and you've got organisms that are trying to kind of balance those adaptations, right? So we're first going to talk about how plants acquire nutrition. Trophy is feeding. That's what the term trophy means. So you can see here autotrophy, photoautotrophy, chemoautotrophy. So how are they feeding? You know, where is their energy source? And so autotrophy means self. They're feeding themselves. So these are like our plants or cyanobacteria, blue-green algae that are able to kind of photosynthesize and make their own sugars. Heterotrophy is an organism that can't make its own food, right? It can't feed on its own. It needs to eat something else. So these are like our carnivores or our herbivores, you know, the elk, the moose, the deer that are having to consume the plants to get their nutrition, or other animals, right? Photoautotrophy means that the energy source is going to be the sunlight, right, in order for it to convert and create its energy. And then chemoautotrophy would be, you know, there are areas where there's not a lot of sunlight available, and there have been some plants and other organisms that have been able to adapt and use other energy sources, so hydrogen sulfide and other gas, in order to do that. So they're gaining energy that way. So again, be aware of what those terms mean, how they're getting their, food. Are they getting it on their own through photosynthesis? Are they getting it by eating other organisms? And then what is their energy source? So the chemo is the chemical, the other kind of, inorganic molecule, and the photo is the sun. Okay. I did put a couple pictures up here, and in part, some of them are just really cute, right? You can't overlook this, leaf sheep, sea slug, right? I did that because more and more research is coming out that there are certain animals that appear to photosynthesize during certain scenarios, like certain environmental conditions that this happens. Early on, it started off, with these sea slugs, so there are a lot of oceanic, organisms seem to kind of have this green color. And it turns out that they have a mutualistic relationship with another organism that does photosynthesize. So initially, people just thought, wow, these are animals that can photosynthesize. And then as they learned a little bit more, they realized, oh, they're just in close, living proximity to these organisms, and they have developed this ability to pull in chloroplasts from these photosynthetic organisms. And when, food is scarce, or, their conditions in the water change, they can ramp up a little bit of photosynthesis using those organelles and make it. We see that with this, leaf sheep sea slug a little more commonly. And then we also have learned that we can see it in terrestrial organisms. So if you guys have ever had, roses, and you've gone outside and you've maybe seen these tiny little bugs that are kind of tapping into their stems and sucking up some of the dew, which is like the sugar water. These aphids will, if you look at them in certain times, will be kind of orangey looking. So do you guys remember what the plant pigment, not chlorophyll, what's the pigment that's in carrots? That if you eat too much, you kind of have an orange hue. You guys remember?
Beta-carotene?
Yeah, carotene. So they have carotenoids. So instead of maybe like taking up the, organelles that are going to have chlorophyll A, they're actually able to kind of tap into another pigment. And these guys, when it gets really stressful, will be able to photosynthesize using these carotenoids. And it seems they've evolved that a little differently than some of these ones that share. So there's some, cool science going on that's kind of like, what's the boundary here between plant and animal, right? And I did mention, I think, the term kleptoplastids, right? So they're klepto, like, if you're a klepto, you're stealing stuff. So they're stealing other organisms' plastids. So they're like cells, right? All right, so we left off here, right? The big story today is photosynthesis, and I just want you guys to know some of the big take homes. This isn't a biochem class. We're not in Bio 101. We're going to just be covering some kind of big take home messages. So you'll want to have it, pad and paper to reinforce some of the big take homes. We've got our carbon dioxide that's going to come in, in the plant stomates. They're going to open up those, cells in their leaves, and they're going to pull in that carbon dioxide. Like I said, they can lose water at the same time. And with the help of light as an energy source, they're going to be able to kind of crank through the Calvin cycle and make some sugars, and the release will be oxygen. Okay. So that's kind of where we're going to focus our attention at the beginning of lecture. So you probably heard of photosystem 1. These are considered to be the light reactions, okay? And there's some really, really good diagrams. I know some of you might be more visual learners, others like words, so I've kind of got a balance here, but I like the visuals. If you look at a cross-section of a leaf and you kind of find a chloroplast that's gonna have the chlorophyll in it, there's layers of cells that are there that when sunlight strikes, the electrons that are found there get really jazzed up. They get super excited and they release energy. And that energy is necessary. And the only way that they get jazzed up is if sun strikes the leaf, okay? So it's important, that's why they call it a light reaction. If, we need energy to make our sugars, we need to have that sunlight or some other kind of inorganic molecule like we talked about with the sulfur that will allow us to jazz up our electrons and release energy. And the energy that it will release will be ATP, okay? So I want you to be just thinking about those electrons kind of moving around excited inside this chloroplast, inside what we call a photosystem. And it's unstable, and so the energy gets released, okay?
Dr. Garneau, can I ask a question about that?
Sure.
I don't know if this is beyond the scope of this class or not.
Yeah.
So is that like analogous to like an electron transport chain that you'd see in a eukaryote? Like they have a work function for the chlorophyll molecule. They get enough energy and it knocks the electrons off to go use to drive ATP.
Absolutely. Yep. It is the same exact process. Yep. Definitely. Yeah, so here's again, the words here. Photosystem 1, I want you to think sun striking. It's the light reaction, so you need sun, right? Sun striking your leaf, exciting your electrons, and releasing ATP. But in addition, it is going to create a molecule, I call it a precursor molecule, NADPH, okay? And this will be necessary. The ATP energy and the NADPH are the two molecules that will be needed to crank around the cycle that will make the sugar, okay? So when I say, hey, what's going on with light reaction, you guys are gonna say back to me, well, we definitely need the sun. We're gonna excite electrons, release energy in the form of ATP and a precursor molecule, NADPH, okay? And then those two molecules will allow us, like I said, to make the sugar in this second step, the dark reaction step, okay? So when we say dark reaction, that suggests we don't necessarily need the sunlight for this part, right? The sunlight's provided us with the energy in that NADPH. We don't need this to be happening in the daytime, okay? So again, no sunlight's needed. We just need those precursor molecules, and the outcome of this dark reaction is to make the sugar that the plant needs to live, right? So how it works, and again, I've got a diagram on like the next few slides that will kind of, reinforce this fact, but just some big take-home messages. You've got your plant, it has a pore, a stomata or a stomate, right? It opens up, it pulls in carbon dioxide from the atmosphere. Okay, that carbon dioxide is going to bind to something, and it's going to bind to a 5-carbon molecule called RUBP. Okay, and this ribulose phosphate combined with that carbon dioxide, now the carbon dioxide has a carbon, so it becomes a 6-carbon molecule. Okay, which is totally unstable. For whatever reason, it's not a stable molecule, so it immediately breaks into 2 3-carbon molecules. Okay, so carbon dioxide comes in from our stomates, it binds to RUBP, becomes very unstable, and converts into this 3-carbon molecule PGA. Okay, it goes through one more conversion to another molecule G3P, which will make the sugar. Okay, again, this will all be seen in this next slide. Now, what catalyzes, what makes this reaction happen, which makes the sugar, is an enzyme called Rubisco. Now, I don't know about you guys, but I think that vanilla wafers are one of the best cookies in the world, along with fortune cookies. And so I always remember Rubisco because of, Nabisco that makes one of my favorite cookies, right? So if you guys can remember that the enzyme is Rubisco, sounds kind of like Nabisco, right? And that the, molecule that the carbon dioxide is going to stick to kind of sounds like how it starts, RUBP and RUB for Rubisco, right? you can kind of make that connection in your mind. So you get an enzyme, Rubisco, catalyzing a reaction where carbon dioxide binds to RUBP, okay? Breaks up into a couple of, it's unstable at this point, 6 carbon molecule unstable, breaks up into a couple smaller ones and then makes sugar, okay? That's really what we're talking about. And none of that can happen without the ATP and the NADPH from our earlier light reaction, okay? So this, again, just the same kind of words. This is the Calvin-Benson cycle. What we just talked about is the Calvin-Benson cycle for C3 plants. Okay, this the 3 carbon molecule plants that makes the sugar, right? Right, we got CO2 coming in, it binds to the RUBP 5 carbon molecule, totally unstable, breaks into those, 2 3 carbon molecules, the PGA and the G3P. That G3P is going to make us the sugar and recycle to continue the process, okay? Rubisco is the enzyme that does it, okay? But Rubisco is very complicated. Rubisco is probably one of the most abundant molecules on Earth, and it is totally inefficient, okay? The only time Rubisco is going to work the way that it should in fixing carbon and driving this reaction to make the sugar is when you have like mild conditions, not super hot, and good water, good moisture availability. So if you're a plant in the desert, Rubisco is not going to be super efficient. You actually need a helper portion of your metabolism to do it, okay? So keep that in mind. Rubisco, is not really good with pulling in that carbon dioxide and binding efficiently. It ends up like losing oxygen, and that creates a weird kind of chemistry in the leaf, okay? And it's called photorespiration. And so we'll learn a little bit about that in a moment, okay? So high temperatures, right? High temperatures freaks out the plant. They close their stomates because they don't want to lose any more water. And that forces them to not be able to pull in any more carbon dioxide in order to make these sugars. And as a result, if you're not taking in any more carbon dioxide to photosynthesize, you're going to start burning that sugar in respiration, right? And in doing so, you release this oxygen. So in plants that are kind of using Rubisco only, there's this inefficiency where, photosynthesis stops at a certain point and we get this kind of cranking of respiration, and they call it photorespiration, making it not super efficient, okay? So you can imagine if I was a plant and it's starting to get warm, I probably want to adapt a different kind of strategy than what these C3 plants do, right? Because they're not using the molecules efficiently, right? So again, C3 plants are great in probably boreal, in Arctic, in temperate. And then as we start moving into the drier, warmer places, we might start seeing plants that have different metabolic strategies, okay? So that's something to note. C3 plants are good with good moisture and good, temperatures. And again, Rubisco, it's complicated. This enzyme is complicated, right? Very important. It's the molecule that's going to allow carbon dioxide to kind of bind to our molecules, but it does it very inefficiently, right? So for every, it's only binding 3 carbon dioxide molecules every second. It's not really efficient. And so as a result, the leaf actually ends up having to have lots of Rubisco molecules inside of it. So they've actually done studies where they've kind of dried the leaves and been able to determine that about 30% of the dry weight of the leaf is made up of this Rubisco molecule. It needs to just pack it in order for it to be able to crank in, pulling in the carbon dioxide and making the sugars, right? So again, not super efficient, but it must do something okay because it still sticks around, right? Yeah, Elliot, what's up?
Yeah, another question. So there's got to be some reason for it being that inefficient, right? Because there's billions of enzymes that are
Yep.
very, very, very efficient. So there had to have been selective pressure for it to be slow.
Exactly.
Any idea what the deal is with that?
Yeah, I think likely it's a function of how our climate has changed over time. So we're going to see some visuals of like maps that show like coming in from the south part of the US, we have a lot of C4 plants. You'll see zones. And then we get up to kind of where we are, and you'll notice the C4 plants kind of taper off and the C3 plants come in. So I think probably, and you'll notice every plant still has this carbon Rubisco RUBP portion of photosynthesis. But when we get to these warmer conditions, we're going to have another step right before it. Okay, so I think just as places were, you know, getting warmer and conditions were changing, they had to have an additional step to become efficient enough to survive in these conditions. So I think, you know, maybe in earlier times, you know, the climate was so different that this was okay. This was the original kind of process, and then they just, over time, as conditions shifted, they had to kind of build those new steps in. So yeah, we'll see that in a sec, but just keep in mind, it's action packed. A leaf is action packed with this enzyme Rubisco. It's not super efficient, right? In part because it's not good with oxygen, right? We have a buildup of oxygen in the leaf, so we're not using, you know, the carbon dioxide as efficiently. But it does help us to fix the carbon. So here's what I was hoping to get to, right? So we've got a cross section of our leaf, right? Imagine this is like you know, the leaf right here. And this would be maybe an ash leaf, okay? And we clipped it and we looked on the microscope at the kind of short end, right? We're looking at, you know, the surface cells here, the epidermis, maybe waxy a little bit, right? And we've got our stomata, right? These are the cells that will open. These are called guard cells. They kind of are on either side, and they can open to pull in carbon dioxide and also release water, right? If that's what's needed to be done. So the photosynthesis steps are going to occur in the mesophyll cells for these C3 plants, okay? And so just imagine the process. This is all we talked about, right? We've got chloroplast right over here, right? Sunlight is striking the chloroplast, getting jazzed up those electrons, right? They are unstable. They'll release their energy as ATP and a molecule NADPH. Those are the precursor molecules. Whoops. And it heads on over to kind of fuel this cycle. This is the Calvin cycle, right? Carbon dioxide comes in from our stomata, binds to that RUBP, right? That 5-carbon sugar. And that binding, that reaction is catalyzed by the Rubisco, right? We remember RUB on Rubisco, okay? It becomes a 6-carbon molecule at that point, right? Which is unstable, so it breaks into these 2 molecules of PGA, this 3-PGA, and then shifts over to this G3P, which will result in our sugar creation and a recycling to kind of keep this fueling, right? It'll just keep going. As long as we have carbon dioxide coming in and an energy source from our light reaction, the cycle will just keep cranking, okay? However, just not super efficient unless you're in those ideal conditions for those C3 plants. All right, so just another beautiful reaction here, right? Light reaction, getting everybody jazzed up, and the ATP and the NADPH go over here to the Calvin cycle, carbon dioxide coming in, binding with the RUBP, breaking down, sugars released, right? That's what we want to make, and it cycles again, okay? All right, moving forward. So that is the C3 metabolic process, right? That's how C3 plants, those 3-carbon molecule plants, we think of the G3P and 3-PGA, right? Soil moisture and temperature. So like our environment plays a role in how well these plants can make sugars, right? And so if it's hot out, no plant's going to want to open those stomates, right? Because they'll lose more water. So they've got to kind of think about what to do, right? And adjust their kind of timing and so on, right? So stomates will close or open depending on those conditions. Here's just an example. Look at the root to shoot ratio of these grassy plants. These are like sand dune plants, so we know we're in a dry area, okay? And so when it's wet, okay, when it's wet, notice the blue, any condition where it's wet, we tend to have less roots, right? This is the root to shoot ratio. So the lower your bar, the less roots. So when it's wet, you're going to shift your energy away from roots to the above ground, right? We learned about this and we had it on the quiz, right? When it's a wet condition, you tend to have more above ground biomass. When it's dry, you tend to have more root biomass underground. So in a dry scenario, these organisms can shunt their energy, their growth, their tissue to the underground, right? So that they can pull up more water. And that's why you'll see that there's greater rooting depth or greater rooting biomass when conditions are scarce. So these plants are kind of flexible in where they can allocate their energy to building structure. Okay, now let's think about an adaptation. It's starting to get warmer. Okay, maybe we're moving towards, you know, grasslands or chaparral. Maybe we're moving into the savanna. Okay, we're going to have higher rates of water loss, right? When the stomates are open because it's warmer, right? It's just going to get pulled on out. But we know we have to take in the carbon dioxide, so we got to kind of risk it, you know? So, you know, when we shut the stomates to not lose the water, we can't take in carbon dioxide and we can't make sugar. We can't grow, right? We can't photosynthesize. A lot of our plants are going to even drop their leaves. They'll be drought deciduous, right? So that stinks, right? Now they can't excite their electrons, right? They can't make the ATP to do this. So here's where that adaptation to developing an additional step kind of comes into play, okay? So we've got C4 plants, so that's a 4-carbon plant, and these CAM plants, that's the crassulacean acid metabolism plants, okay? These are gonna be plants that deal with heat better, okay, and water reduction, all right? So the C4 plants are actually going to have 2 different locations in their cells where the photosynthesis will occur. And that's going to help them to be a little bit more efficient. And the CAM plants, which are like our desert plants, the succulents, the cacti, right, the yucca, they're going to actually, instead of like photosynthesizing in 2 different locations in the cell, they're going to do it at 2 different times, right? What they're going to do, which is totally smart, right? Think about being in the desert. They're going to open their stomata at night when it's not really hot, so they lose less water. And they'll pull in the carbon dioxide at night when they're going to lose less water, and they'll store it as an acid that can then, in the light of the day, when the ATP can be created, will move through that cycle like we learned earlier and crank out the sugars, right? It's a pretty smart strategy for them. Okay, so let's just talk about the C4 pathway. And I'm going to have, again, great visuals if you're more of the visual learner. So instead of starting with the CO2 binding to the RUBP, right, catalyzed by Rubisco, like we saw with C3, we're going to actually have a new carbon molecule, PEP, okay, that will bind with carbon dioxide. And it's going to have to be catalyzed by a different enzyme, okay? Luckily, it's an easy one to remember. It's PEP carboxylase, okay? Why is this a good thing? It doesn't have that weird reaction with the oxygen, right? It is much more efficient in these warmer temperatures. We see less photorespiration occurring inside the leaves, okay? So again, C3 plants, RUBP, we get this weird byproduct, right? This photorespiration problem, making those plants 25% less efficient, right? So, you know, not ideal when they are stressed by water and higher temperatures, those C3 plants. So what they've done is they take in that carbon dioxide, they bind it to the PEP, and as I said earlier, they're going to convert it into this acid and shunt the acid down into a new location in the cells. For whatever reason, I'm not a plant, a botanist or anything. For whatever reason, shunting it to this other location is another, way that they've made this a little more efficient for them, okay? So much more efficient in photosynthesis, not having that weird binding issue with the oxygen, the photorespiration, right? Not being 25% less efficient, using that carbon dioxide more fully. And it happens in areas, again, where the soil's drier and where it's a little bit warmer, okay? So again, more of the words, right? Carbon dioxide coming in, binding with the PEP, converting to an acid. That's all you need to know. You don't have to memorize these acid names, okay? Converts to this oxaloacetic acid. And it does it in the mesophyll cells, which is the same place that we saw that Calvin cycle happening in our C3 plant. So the mesophyll cells are where this kind of fixation tends to occur, okay? And so we've got, PEP carboxylase, which is the enzyme that has to make this thing happen. We can close our stomata so that we don't lose the water. We have an enzyme that's much more efficient, so that's not a major problem. And over time, that acid will break down and trickle in carbon dioxide And we can shunt that carbon dioxide into another cell. So it'll leave the mesophyll cell, go into the bundle sheath cell, and we can run through that Calvin cycle, okay? So C4 plants, again, just take home messages here. They've evolved a way to not have that photorespiration issue, right? They're more efficient with the carbon dioxide. They will move their carbon molecules, in from the mesophyll where the PEP and PEP carboxylase stuff is going on over into another cell, a new cell, the bundle sheath cell, where it'll go through the Calvin cycle, okay? Just like we saw with the C3 plants, okay? So let's take a look in a sec at what this looks like as we kind of compare across them. But one of the things I wanted to say before we jumped over is that, we know that this inefficiency with Rubisco is a problem. And there are a lot of people out there, that are doing some genetically modified experiments, right? So they're genetically modifying a lot of these plants to make it so that Rubisco is a little bit less, efficient. That whole process is a little bit less, is more efficient, right? So here we've got 4 unmodified plants on the left, and we've got 4 genetically modified plants over here on the right, okay? And it has boosted productivity by 40%, right? So, what they're doing, and that this was, crops like rice and wheat. So again, think about our bread basket. If we could plant crops like this and have them be more efficient with greater yield, right? So Rubisco, right? It's trouble dealing with carbon dioxide and oxygen as it gets hotter. We get that buildup of oxygen, photorespiration, right? And so their goal is to build these better plants so that they can deal with these changes in temperature that we're expecting we're going to be seeing. So we're going to have to be on the lookout. There's perhaps going to be some more genetically modified plants to kind of help deal with this. So again, how does this look, right? We got a cross section of our plant. We've got our mesophyll cells, just like in the C3 plants, right, where the photosynthesis was occurring. We also have an additional row of cells. These are the bundle sheath cells, okay? So let's take a look at what happens. In the mesophyll cells, carbon dioxide is coming in and it's binding to the PEP, okay? That PEP molecule will take on that extra carbon from the carbon dioxide with the help of that enzyme, PEP carboxylase. Way better to deal with the higher temps, okay? And it's going to convert into this acid for long-term storage. The acid will scoot on over to the new cell, the bundle sheath cell, and trickle in carbon dioxide that will drive that Calvin cycle. Remember the Calvin cycle, right? Carbon dioxide comes in, sticks onto the RUBP, catalyzed by Rubisco, breaks down into PGA and G3P to make the sugar, and recycles itself. Okay, so what we've just seen is these C4 plants in warmer places, drier places, have an additional cell. They will start off the process of photosynthesis by taking in carbon dioxide into the regular mesophyll cell. They will take that CO2 molecule and tack it right onto that PEP, okay? The PEP and carbon dioxide, that reaction happens with PEP carboxylase enzyme, converts it into an acid for greater long-term storage. The acid will shift into the bundle sheath cell and will trickle off. It'll kind of like release the carbon dioxide off the acid. And it will feed into the Calvin cycle, right? Where CO2 is going to bind to your RUBP, catalyzed by Rubisco, and then we pump out our sugars, okay? So remember, C3 plants, everything's happening in the mesophyll cell. C4 plants, we've got 2 spots now. We've got the new thing going on in the mesophyll cell, and we've got our typical kind of Calvin reaction where sugar is happening in the bundle sheath cell, okay? Who are these C4 plants? Well, it's important to note, if you look over here, we don't see very many woody plants. So drier conditions, right? Less, when you have more water, you have more woody plants. Okay, so we tend to see kind of the herbaceous plants that are C4 type plants, the grasses, the wetland grasses, the marsh grasses, the crops. Okay, there are some trade-offs. You have an extra carbon fixation step. That takes some energy, right? to make that happen. We have more enzymes. We have the PEP carboxylase as well as Rubisco, and you're going to need maybe a little more structure, right? We have the bundle sheath cell that we have to build in there, and that's going to take some energy as well, okay? However, it balances out. It allows them to do well and to get by and to have increased biomass in areas where it's drier and where it's hotter, right? And so again, we see, you know, herbaceous plants maybe have less tissue, less structure. It's easier to build with this kind of energy constraint that they may have. Less above ground growth in leaf tissue, and not necessarily, they're kind of not necessarily giant plants like a tree would be, right? They're more of our grassy stuff, which is smaller. Okay. And so I did mention kind of, you know, Elliot had mentioned, you know, why on earth would we even have C3? It seems totally inefficient. Like, why would that have evolved? Well, again, look down here at this map. These zones are showing you the percent of C4 like grasses that you might find if you were in parts of these countries, right? So if we're up kind of where we are, we may not see any or very few C4 grasses, but as we get into drier, warmer places, we start seeing 60% of the plants, you know, 80% are going to be of the C4 type, okay? And we can go over here and we can look at elevation too, right? Proportion of C4 plants as a function of elevation. So low elevation where it might be drier, we see a greater proportion of C4 plants. As we get to higher altitudes, we don't see as many of those C4 plants, right? It's just like how we would expect, you know, we've got latitudinal gradient, we've got an elevational gradient. It goes from hot, it goes, you know, as we move up in latitude, you know, up here to the northern latitudes, it's going to be, you know, fewer and fewer of them. We go from hot at the bottom of a mountain. As we go up the mountain, it gets cold. We would expect to see fewer of them as well, okay? And you can see the plant has an adaptation, right? The plant will photosynthesize. You think about the temperature that it can tolerate, right? We all have a tolerance for, you know, certain conditions. C3 grasses don't have, you know, C3 doesn't have a wide temperature, you know, gradient, right? It's not, it can't handle the heat, right? So you see them from kind of 10 to 30 degrees. C4 grasses can shift a little bit more into the warmer, right? C4 and desert shrubs and even cam plants will shift even farther into the more hot conditions, right? They can tolerate that a little bit. All right, we're going to jump right over to the cam plants. Okay, these are the plants that are in the desert, you know, driest, hottest conditions that you can expect. They're going to do something a little bit different as well. Okay, these hot areas, the plants will have a metabolism, a photosynthetic process that is this cam crassulacean acid metabolism, right? They're going to be the cacti and stuff. They're going to do this a similar thing. They're going to use sort of the, you know, PEP carboxylase and PEP, turn it into an acid scenario, and then crank it through the Calvin cycle, but they're not going to do it in a different location. They're going to do it all in the mesophyll cell, okay? But what I did tell you about these desert plants is they have to do it at different times. So instead of in different places, different cells, bundle sheath versus mesophyll, they're going to do it at night versus day, okay? So always in the mesophyll, but just different times of the day. And these are usually not very productive plants, okay? So let's take a look, right? Now, instead of this being like, you know, here's the mesophyll, here's the bundle sheath cell, this is all in the mesophyll cell, but at nighttime, CO2 is going to come in, it's going to bind with the PEP, right? Stomates will open at night when they're not going to lose water. It's going to bind with the PEP molecule. PEP carboxylase is the enzyme that makes that happen, converts it over to an acid. The acid will then trickle carbon dioxide, again, all in the mesophyll, down into the Calvin cycle, right, where it's going to bind with the RUBP, catalyzed by Rubisco, right, and immediately break into those other 3 carbon molecules, releasing the sugars, right, and cycling again. It will do this in the daytime. So the Calvin cycle will happen in the daytime. Why? What does it need? How do we crank this?
It needs light.
Needs light, needs the light to make the energy, right? Needs the light to make that ATP to get that going in the NADPH, okay? So again, stomata are closed, those pores close at night to not lose the water when it's dry and light. and they'll pull in the carbon dioxide then at lower risk and work it very similar to what we saw with our C4 plants. So now this is kind of the pièce de résistance, right? This shows us everything kind of laid out. Our old school C3 plants, moderate temps, cold or moderate temps, temperate forest, taken in CO2, binding to the RUBP, Rubisco, the enzyme catalyzing it, breaking down into those 3 carbon molecules, releasing sugars, okay? We go over here to our C4 plants, the grassier types, right? We've got 2 different cells now, mesophyll and our bundle sheet cells. Spatial separation, they're separating the steps of photosynthesis out in space. Carbon dioxide coming in and binding to PEP, catalyzed by PEP carboxylase, converting it to an acid. The acid will kind of trickle into our bundle sheet cell, spilling out carbon dioxide, binding to your RUBP, catalyzed by Rubisco, breaking down into those 3 carbon sugars and, breaking down into the sugars and recycling, okay? Only difference here is in the desert plants, it's all happening in the mesophyll, just like it was in the C3 plants, just different times a day. So temporal separation, okay? Open the stomates at night, pull in the carbon dioxide, go through the PEP process, right? Make it into an acid, trickle the acid in, make your sugars, okay? So definitely check that out. And I think this is really cool. It ties into our biomes nicely, right? So look over here at your tundra, picture of your tundra. No trees, low-lying, prostrate, organisms growing low to the ground with roots, mostly C3s, all C3s, right? So it's cold and dry, right? Now we go to taiga, maybe a little warmer, maybe a little more wet, still C3s. Now we go over to the temperate, okay? Temperate, broadleaf and conifers, temperate deciduous, still C3s, okay? Now we're going drier, warmer, starting to get more grasses, savanna, C4s, right? Tall grass prairie, C3s and C4s. C4s, prairies, right? Chaparral, getting drier again, C4 to cam. And then deserts, mostly cam, okay? So it's kind of neat to see how that would shift when you think about it from that biome perspective, right? Now, all of these organisms, once they've made their sugars, right, need to use it, right, in order to grow, to repair, to, make seeds. And so, they will burn that sugar in respiration, right? Plants and animals respire. And so you can see the sugar here with the help of oxygen will break down into carbon dioxide and water, releasing some of that ATP. And it will do that kind of, in the presence of oxygen, they call that aerobic respiration, right? Aerobic with oxygen. And then anaerobic is also known, that kind of respiration is also known as, fermentation. You probably, thought of like beer and yeast and fermentation that way, but also, the runners and the buildup of the lactic acid in muscles when they're running and they're oxygen deprived, musculature and so on. So again, plants and animals respire. Everybody's got to burn that sugar in order to survive. So there's limitations to photosynthesis. We've talked about like water and temperature, right? Limiting photosynthesis. Think about light though, okay? Think about a plant exposed to extreme light. You guys might have seen this. It might change color, right? It might shrivel up. Might get spots. So, net photosynthesis in terms of irradiance, how bright something, strikes it. When there's not a lot of light, photosynthesis is not super efficient, right? It doesn't have a ton of, available excitement in the electrons, right, to release the energy. At a certain point, when it gets, bright enough, we can saturate the leaf and you can have maximum photosynthesis, right? You can have this light compensation point where photosynthesis equals respiration and they're kind of happy. But then at a certain point, if it gets too bright, the plant's going to shut itself down and it's not going to photosynthesize anymore, but it will continue to respire. And that creates bad conditions like we talked about, that photorespiration scenario that we talked about. Shady forests have low photosynthetic rates. So you can imagine the plants either need to have tons of Rubisco in their leaves or big leaves or, some other way to deal with not being able to photosynthesize very well, okay? Carbon dioxide, same sort of thing that we saw with our light levels, right? At a certain point, there's too much carbon dioxide and it kind of shuts itself down. So again, stomates that are opening to pull in the carbon dioxide, but also can release the water. It's kind of important to think about the levels of carbon dioxide in the atmosphere, and how that might change the opening and closing of the stomates, not just the light, not just the water. Okay. So again, we want to open the stomates to get the carbon dioxide. If we don't, we're not going to make sugars, but if we do, we might lose our water, and that can be a problem. So I told you earlier, we've got these stomata, and there's 2 cells on either side of those pores. They're called guard cells. And if there's a lot of water, they're going to swell, those cells swell up and shut. Okay. And if there's not a lot of water, they'll kind of release themselves a little bit and they can open. This idea of kind of swelling up and kind of, releasing is this turgor pressure, right? Forcing, moisture into these cells, making them rigid, right? A plant will grow the best when it has enough water to be rigid, to be turgid, they call it, right? A plant that you forget to water a couple days in a row is just not going to be photosynthesizing at its maximum. So we have to keep in mind those things, right? If you want a plant to be happy and growing well, you need to make sure that it's watered so that it can maintain that structure. And what kind of impacts their ability to stay hydrated and be rigid like that is also the atmosphere, right? If you think about walking outside when it's really humid and muggy, and you're a plant trying to open your stomata to take in carbon dioxide, if it's really dry out, it's gonna just wick water right out of the leaf. It's just gonna pull it right out, moving it right from the roots, root hairs, on up through the plant, pulling it up through capillary action and so on, and then the wind that's pulling it off if it's dry out. But if it's warm out and muggy, it's saturated already, so it's not going to pull that water out. Okay, so there, depending on your conditions outside, it might affect whether or not your plant is going to be turgid or not, right? And it can affect how well it's going to photosynthesize. Also, the type of soil that the plant is in is going to, think about how the water is going to be held at the roots or, leach down away from the roots. So again, you want your plant to have good amounts of water. And so you'll see the visual here, clay soils versus sandy soils. And I have talked about this before. Sandy soils are big and high surface area to volume ratio, and the water just trickles right out. And the clay soils are going to have, an ability to hang on to the water. So look over here at the water content in the soil of this is field capacity and, the wilting abilities of sand and silt, right? So you're not going to have a lot of water in sand. When you get over to loam, you're going to have a little bit more. When you get over to clay, you have the maximum. So a plant in clay is going to have better access to water than one that would be found in a sandy soil. Again, think about the desert plants. And that's partly why they adapt that strategy to open stomates at night. Okay. Now, how do the plants that are floating in the water take in carbon dioxide? Well, if they're like lily pads or anything that you can see outside of the water, they're going to have stomata, and they can do it the same way. They can just open it up, pull in the carbon dioxide, and go ahead and make their sugars, right? But if they're underwater, they actually have to take in carbon dioxide in the process of diffusion. So they'll take bicarbonate, HCO3, instead of carbon dioxide, they take this bicarbonate and it diffuses across their cell walls underwater, okay? And that's how they take in the carbon that will crank the photosynthesis process. So keep that in mind. Totally submerged underwater plants get their carbon not like carbon dioxide, but diffused across their cell walls as bicarbonate. The ones on the top of the water can still go through the same process. All right, you guys, Monday we will finish up. We only have a few more slides about morphology of the plants. So we'll finish up plants and we'll start on our animals on Monday. If I were you guys, I'd get a jump on studying, right? We want to go through lesson 1, review the quiz, lesson 2, the biomes, maybe do the biome Jeopardy as a way to quiz yourself. Lesson 3 is the limits to organismal distribution. Remember the circulating air masses and other organisms kind of limiting where you are. And lesson 4 is our plants, okay? So make sure you review, at least on the test, will be up through plants. So make sure you guys start using some time this weekend to begin your review, okay?
Thank you.
You're welcome. Have a good weekend, you guys.
Thank you.
Yep, see ya.
Thank you.
Bye.
Yeah, have a great weekend.
Bye.